Again for the NEET classes, and very important topic we are discussing is transformation and what is the purpose of this transformation. So transformation is a method that we are introducing the foreign DNA in the bacterial cell. That means if we have the foreign DNA that we want to introduce into the bacterial cell, we need to perform this process that is called as a transformation, and it's also called as the calcium chloride transformation technique. Chloride transformation technique. You need to remember the second name of this. So first of all, we will try to understand what is competent cell and what do you mean by competent cells. So the competent cells are the bacterial cells that can accept extra chromosomal DNA, that is naked DNA. So we now try to understand competent cells. So they are the bacterial cells. They are the bacterial cells that can accept extra chromosomal DNA, that can accept extra chromosomal DNA. So this extra chromosomal DNA is all called as a plasmid, and you need to remember it is a naked DNA from environment. So the process of generation of competent cell may occur through the two methods. One is natural competence and another is artificial competence. So there are two types of methods of making the competent cell. One is called as a natural and another is artificial competence. So the competence can be developed by the two ways. One is natural competence, another is artificial competence. So first of all, we will try to understand what is natural competence. So the natural competence is a genetic ability of bacterium to receive the DNA from environment under natural or in vitro conditions. So this is a natural competence. So the bacteria is having the natural competence to receive the foreign DNA from the environment. That is natural competence. Natural competence. So natural competence is when bacteria genetically, it is the genetic ability, when bacteria is having genetic ability to receive DNA from environment. So this is a genetic ability or natural ability to receive DNA from environment. So but it can be done by the natural way or in vitro way. Bacteria can also be made competent artificially by making them treatment and a heat shock to make the transient permeable to DNA. That is the bacterial cell. The bacterial cell is having the bacterial cell wall outside it and the cell membrane. This is a cell wall, and this cell wall is made of lipopolysaccharides, and this is a cell membrane. So this cell wall and cell membrane, this foreign DNA need to enter both of these. We can't make the pore artificially inside this bacterial cell wall and then introduce the DNA. So we need to develop a condition through which this bacterial cell wall will facilitate, and this bacterial cell membrane will facilitate entry of the DNA into the bacteria. So how can we make this bacteria competent to get this foreign DNA? That is called as the process of formation of competent cells. So that can be done by artificially. Naturally, I told you, is the genetic ability of the bacteria. Artificially, how can we do it? So artificially we can do it by giving the heat shock or a chemical treatment. So we will give the chemical treatment. we will give the chemical treatment, and we will give the heat shock. We will give the heat shock, so followed by heat shock. So then, bacteria can also made competent by the chemical treatment and heat shock. And natural competence date back 1928, when Griffith discovered that the prepared heat killed cells of the pathogenic bacteria would transform the non-pathogenic cells into the pathogenic type. What is the example of this natural competence? Is the Griffith's experiment, where you have seen that when Griffith growed the pathogenic heat killed pathogenic bacteria and non-pathogenic bacteria together, and they heat killed non-pathogenic bacteria developed the pathogenic, become the pathogenic type. And natural competence has been reported in many bacterial strains like Bacillus subtilis and Streptococcus pneumonia and Neisseria gonorrhea and Hemophilia influenzae. So there are types of the bacteria who are having the natural competence. So I will write down what type of strains of bacteria are having the natural competence. First of all, it is Bacillus. It is Bacillus subtilis. Another is Streptococcus coccus. It is Streptococcus pneumonia. Pneumonia. It is Streptococcus pneumonia. Another is Neisseria gonorrhea. Neisseria gonorrhea, and another is Hemophilia. You can note down these things. Hemophilus influenzae. They are the type of bacteria who are having the natural competence or a genetic competence. Natural competence is highly regulated in bacteria and varies across the genera. It is a regulated process and it varies across the genera. And in some genera, certain portion of the population are competent at a time, and in other, the whole population gains competence at some time. When the foreign DNA enters inside the cell, when the foreign DNA enters inside the cell, it may be degraded by the cellular nucleases or may combine with the cellular chromosome. So, if the this foreign DNA enters inside the cell, it can be degraded by nucleases or it can combine with the DNA. It can combine with the DNA. It can combine with the DNA. So, and this is the possibility that nucleus may degrade them or combine with the DNA. However, natural competence and transformations are efficient for linear molecules such as chromosomal DNA, but not for the circular plasmids. If this is the natural competencies for the linear molecules. This is very important point, not for the circular DNA. This is the natural competencies only for the linear molecules. That if the DNA is a linear, then it will go introduce, it will be received by the bacteria and not for the circular DNA. So but not for the circular plasma. Artificial competence is not coded by the genes or bacterial cell. It is a laboratory procedure in which the cells are passively made permeable in the DNA using unnatural conditions. So now there is a question that if we are having the plasmid which is circular plasmid, how it will enter into the bacterial cell? For that, we go for the artificial competence, and we perform this by using a unnatural conditions. So we go for unnatural conditions. Unnatural conditions. So what are unnatural conditions is by artificial competence. Competence. So this is very important. Why is artificial competence needed if there is a way or a process of having the natural competence? So there is another drawback of the natural competence, that natural competence only receives the linear DNA, not the circular DNA. So this is very important to understand. And uh, this type of knowledge you will rarely get any other video, because they are not used to give the detailed information to you. So we will try to understand what is the principle of the competence. So the principle of the competence is that we go for the plasmid DNA, and then we have the E. coli, and we want to introduce this plasmid DNA into this E. coli, and it is carried by using the ice cold calcium chloride. Now we will try to understand what is the purpose of using this ice cold calcium. Why we can't use sodium chloride? Why we can't use potassium chloride? Why we use this ice cold calcium chloride? Now it is very important to understand that DNA is highly hydrophilic molecule. It is hydrophilic. We will first understand the properties of DNA. So DNA is hydrophilic. Hydrophilic molecule. Normally, it cannot pass through the cell membrane of bacteria. It will not pass through the cell membrane of the bacteria. That because of the hydrophilicity, it will not pass through the cell membrane of bacteria. So DNA is hydrophilic. It will not pass through the cell membrane of the bacteria. Hence, in order to make it to enter the get internalized by the bacteria, they must be made competent. That means we need to make these bacterial cells competent. So we need to make them competent. We need to make them competent. So how can we make them competent? What is the way of making them competent? This can be achieved by making a small hole in bacterial cell by suspending them in a solution containing a high concentration of calcium. So this why we use the calcium. It makes the small holes or small pores or small holes, so that this bacteria which is a hydrophilic will enter into the bacterial cell. So we need to develop this here. So extra chromosomal DNA will be forced to enter the cell by incubating the competent cell and the DNA together in ice, followed by the heat shock, and that causes the bacteria to take up. That is, we have to put it into the ice and then followed by the heat shock, and which will help it to enter the bacterial membrane or a cell membrane. And the bacteria no longer now the bacteria no longer becomes stable when they possess the hole on the cell, so may die easily. Additionally, a poorly performed procedure may lead to enough competent cells to take up lead to not enough competent cells to take DNA. So it has been reported that naked DNA molecule is bound to LPS, lipopolysaccharide receptor molecule, on the competent cell surface, and divalent cation generates a coordination complex with a negatively charged DNA a molecule and LPS, and DNA becomes large molecule and cannot itself cross the cell membrane to enter into the cytoplasm. So heat shock step strongly depolarize the cell membrane of calcium treated cells, and thus decrease in membrane potential lowers the negativity of the cell membrane inside the inside potential, which ultimately allows the movement of negatively charged DNA into the cell interior, and subsequent cold shock raises the membrane potential to its original value. Now we will try to understand what I told you first. Now we will try to understand in a step by step manner and keep watching what we have discussed. So the question is, the DNA is hydrophilic. The DNA is hydrophilic. Hydrophilic. The other property of DNA is backbone of DNA is negatively charged. Third thing is the cell membrane of this, yeah, cell wall of this bacterial cell, that is LPS, lipopolysaccharide membrane, is also negatively charged. So what is this work of what this calcium chloride does? This calcium chloride, when we put this bacterial cell into the calcium chloride, that is ice cold calcium chloride, it neutralizes the DNA. It neutralizes the negative charge of DNA by calcium 2 positive. So it causes the neutralization, and it also neutralizes the LPS, and it also causes the DNA adhesion or binding on LPS membrane, DNA binding on the membrane, binding on membrane. These are the three things it causes. And why we give heat shock? Now this becomes the large DNA. It is the DNA attached with the calcium, and it is a very large size. We give the heat shock. That means the membrane is depolarized. That means inside membrane is now depolarized, and this depolarized membrane easily allows the DNA, naked DNA, to enter into this cell. And then we will again give the ice cold shock. That means we will repolarize the membrane. Polarize the membrane. We will repolarize the membrane. So it is very important. So you have understood what is the purpose of giving this, using this calcium chloride. Now another thing is, so calcium chloride transformation technique is most efficient technique among the competent cells, and adding calcium to the cell suspension allows binding of DNA to LPS. That I told you that it allows, that helps in the binding of DNA to LPS, lipopolysaccharide membrane. Thus, both the negative charge of DNA backbone and LPS can come together, and when heat shock is provided, plasmid DNA passes into this membrane. And what is the concentration of calcium? It is zero point 0.1 molar concentration of the calcium which should be used. So what is procedure, or what is the method? How can we perform this in the laboratory? That is very important, and that we need to understand. How can we perform this in the laboratory? So what is the mechanism, or what is the standard procedure of doing this? Very important topic, and very important to understand this whole process of transformation. If you are working in the laboratory, are you going to join the laboratory, or you are working for the examination, there are very few important questions which will be sometimes asked in the interview, or very sometimes you will face, or will create the confusion. So first of all, we need the bacteria. To to need the bacteria, we will grow this bacteria. That means we will go for the culturing of bacteria. Culturing bacteria. So the bacteria that will be cultured will grow. How do we know whether the bacteria has grown or not? And this bacteria is having the growth phases. One is called as the lag phase, another is called as the log phase, and third is stationary phase. So for making the competent bacteria, for making the bacteria competent for taking this naked DNA, we need the bacteria from log phase. How do we know whether the culture bacteria is in the log phase? We will check the OD. The OD should be between zero point 0.2 to 0.4 at 550 nanometers. This is the wavelength. At 540 nanometers, this is the wavelength. Now we will take this bacteria. We will take this bacteria inside this culture disk, and we will put the ice below this, so that we will give the treatment that is called a ice treatment. So first is growth, second is ice treatment. second is ice treatment, and third is ice treatment is kept, that is cells are kept on the ice to stabilize the membrane. Stabilize membrane. Now another is calcium calcium chloride washing. Chloride washing. Calcium chloride washing, that is CaCl2 washing, and you know that the concentration should be 0.1 molar calcium chloride, and the calcium causes the binding to the DNA, and it causes the binding to the LPS, and following that we will put this into the incubation period. That means we'll allow them to bind the naked DNA and to bind the LPS. That means to neutralize the negative charge of the DNA and neutralize the negative charge of the membrane, and then followed by the heat shock. Then we will put the naked DNA into it. Now the naked DNA will be put into it, and the competent cells, oh, the calcium will bind to this naked DNA and it will bind to the LPS, and then followed by the heat shock. Heat shock. Followed by the heat shock, and this following by the heat shock will it be for the 90 seconds, and it will cause the pore opening by depolarizing the membrane. Polarizing the membrane. It will cause a depolarization of the membrane, and this is the process of transformation. Now you might have understood what is the transformation and what are the methods of the performing transformation very easily. And now we will move to the another topic, that is conjugation. And first of all, I will clear this whiteboard. If you have any doubt, you can ask the questions. You can comment below. If you have any question, you can ask them. And if you want, we can start the 10 days most important question series this time. 10 days most important question series for the NEET at 11 p.m. That means in the night. If you want that to happen, you can comment the yes, and if you don't want that to happen, and we are going to start again that. Now we will understand conjugation. What is conjugation? The conjugation is a natural bacterial recombination process. It is a bacterial recombination process in which the two live bacteria, one is donor, another is recipient, come together and join cytoplasmic bridges and transform the single stranded DNA from donor to recipient. That means two bacteria, and both those bacterias, one is donor, another is recipient. This is positive, this is negative. They form the cytoplasmic bridge, that is that is apile, yeah, pile, and they come together and transfer the, that is they are also called as the sex pile, these cytoplasmic bridges. So they transfer this DNA, and it is they also get this DNA from the single stranded DNA from the other bacteria, that is called as a transformation. So the bacterial conjugation or genetic engineering, this is a form of horizontal gene transfer. This is a form of horizontal gene transfer. This is most important question. What is a type of horizontal gene transfer? In most of the exams this is called asked, and this is called as the horizontal gene transfer, where the donor transfers the genetic materials to the recipient through the direct physical contact, through the direct physical contact. So donor, that is called as F positive, and recipient, that is called as the F negative. So donor and recipient produces a pilus, and this pilus that attach to the recipient and bridges forms and the transfer takes place. What is application of this? The application of this is genetic mapping. Genetic mapping used to determine the order of gene, transgenic plant, and spreading trait. Spreading trait, that is antibiotic resistance. Antibiotic resistance. This is very important. So now another topic, this is the bacterial transformation process, that is competent cell preparation, transformation, recovery, and plating happens. Then conjugation, in which the F positive is transferred to F negative, and transduction is the process in which the bacteriophage transfers the DNA to the bacteria, and vasoduction is the with the vesicles are formed, and these vesicles are transformed to recipient. So this is all for today. These are the two videos. Please share the videos, and in the next topic we will learn about the electroporation and further other topics like liposome mediated gene transfer. And in this very important question, in order to make the bacterial cell the competent, what are the chemicals which are used to form the competent plasmid? And the answer is calcium chloride. So please share this video, and please like and subscribe this channel, and try to spread this information among the others. And we are going to start very tonight at 11 p.m. We will you go for very important multiple choice questions for NEET 2026 every day. Every day. Chapter wise we will go for 1 hour. This is a very important announcement, and please share this video. Okay. Thank you, all of you.